Okay, class, now we want to put it all together, in which we'll call chemical digestion. So we'll do a few videos on this just to get everything. All right, so the key to this chemical digestion is to take the polymer form to the monomer. So let's see what we're talking about here. Chemical digestion. Food must be broken down, called catabolism, down into its monomer form. Take that. To its monomer forms to be absorbed. Okay? So we must break it down to the monomer forms to be absorbed. Okay? And ultimately used for energy, growth, and repair, because that's why you eat. Somebody might say, why do you eat? To produce energy, ATP, growth, or repair. Okay? Enzymes along the GI tract. And remember that GI tract is long. You started the initial enzymes in the mouth, and then they proceeded all until you got to the end of the small intestine. No more enzymes after that time. Small intestine. We're looking at breaking it down to what? The monomer form. So let's look at it. AP1, carbohydrate. What's the monomer? Monosaccharide. What's the polymer? Polysaccharide. Protein. What's the monomer? Amino acid. Okay. Fat, triglyceride, fatty acids. Nucleic acid, nucleotide. But a nucleotide is made up of a sugar. If it's DNA, deoxyribose. If it's RNA, ribose, phosphate group, and a nitrogenous base. Adenine, cytosine, guanine, thymine, and uracil. So what's my point? To be absorbed, it must be in the monomer. You're eating it in the polymer. You're actually eating it more than in the polymer. You're eating chunks of food, big chunks of food, big steak, big this. So it's a bunch of polymers. Think about what I'm saying. I mean, you know what I'm talking about. It's a bunch of polymers, and you're trying to break down all of those polymers, those trillions of molecules down to monomers. So this is what you're looking at. And you need to do a lot of it before it gets to the small intestine, and then finish it in the small intestine, because once it gets into the large intestine, it's waste. So whatever you don't get done before it gets into the large intestine, it's going to be waste. So just keep that in mind now. Let's be literal. All right. To use this to make ATP, what do you need to do? You need to metabolize it, right? Now here's a cell. It's in the cell that you're going to do metabolism. It's in the cell you're going to do metabolism. Okay? So what you're doing is, you're taking these various foods here. Once you get it to the monomer and get it absorbed and get inside some of these cells, and you're taking these proteins, carbohydrates, and fats, and then you're going to burn them in something, in glycolysis and the Krebs cycle. Some of you have heard of this. So inside the cell, you have, this would be inside the cell. Inside the cell, you do glycolysis. This is done in the cytoplasm. Now I'm just showing you some stuff. Just want you to be familiar a little bit with this. Inside the cytoplasm, I'm starting with glucose, but however, the process is going to be, I'll show you what I'm saying. So glucose will go through about 10 steps of conversion. Now, the form that you started out when you ate, if you ate animal food, it was in glycogen. If you ate plant, it was in something called amylose or amylopectin. So the polymer of carbohydrate in plants is amylose or amylopectin, but in animals, glycogen. It's groups of glucose. So you broke it down to the monomer, glucose, hopefully, and then glucose would then come in and then be converted through 10 steps of conversion to something called pyruvate. Now, along this way, you make 4 ATPs, but you use 2. So you only get 2. That's not a good efficiency. The key to this glycolysis is you don't need oxygen. This happens in the cell cytoplasm. This happens quick. So this is your quick energy. You got the glucose by either breaking down plant amylose or amylopectin or animal food like steak, whatsoever, glycogen. And then enzymes broke that down to glucose. That's the enzymes I'm talking about as you go through the digestive tract. Okay? Now, once you do this, which is in the cytoplasm, then pyruvate, if oxygen is available, you hear my word, if oxygen is available, can enter into the mitochondria. And in the mitochondria, you do this here, which is called the Krebs cycle. The Krebs cycle. Now, this cycle here will give you 32 ATPs. It only has 8 steps. 8 steps to get 32 ATPs. But what do you need? Oxygen. What do you need? Oxygen. Okay, so here again, pyruvic acid will enter the mitochondria because it will convert to acetyl-CoA, but what do you need? Oxygen. Okay, so then, here we go, glycolysis, pyruvic acid enters the mitochondria. Here you only produce a few, but you get them quick. Here you produce a lot of them. Now the other thing, the Krebs cycle hands stuff to something else in the mitochondria, known as the electron transport chain. Remember, I brought this up in respiratory. And the electron transport chain is really where you cash in the material for the Krebs cycle. That's why I said the Krebs cycle and this are coupled. If we uncouple it, you say, what could uncouple it? One thing is cyanide poisoning. Then you could do all of this and still not get the energy that you required. And so again, what you're trying to do, and here's the electron transport chain. So again, and that's it there. So let me go back. Okay, so what you're trying to do, let's just be sure before we get into this, is you eat chunks of food. The chunks of food are being chewed up in the mouth in the boluses. They're also being worked on by enzymes in the mouth, a salivary amylase and a salivary lipase. The amylase does more. Why do they call it amylase? Because they named it after the plant carbohydrate amylose. Okay? Then what happens is you do some. Then it goes down the esophagus. Esophagus lets you continue with what the mouth did as far as the enzymatic breakdown. It's going down the esophagus in a bolus. Okay? Once it gets into the stomach, then the stomach acid stops all the salivary stuff. So all of that amylase and lipase from the mouth gets stopped because it can't tolerate hydrochloric acid. Okay? So then you're in the stomach. The stomach starts churning those boluses into chyme. But on an enzymatic level, you start working on proteins. Why? In the stomach, you had pepsinogen being secreted from the chief cells. The parietal cells came in, hydrochloric acid, and converted pepsinogen to pepsin, which is a proteolytic enzyme. Once pepsin was made, it acted as an enzyme for itself to make more pepsin. So you start breaking down proteins. Okay? Stomach churns it. What leaves the stomach is a slurry mixture called chyme. Chyme goes into the small intestine, but the small intestine is much narrower than the stomach, so you send about 3 cc's per squirt. And you do about 3 squirts per minute. Remember? Then you go into the small intestine. In the small intestine, that's where you pick up bile coming from the gallbladder and liver, and you pick up pancreatic enzymes. Okay? But what went into the small intestine was partially digested carbs, partially digested lipids and partially digested proteins. That's what went in there. So now in the small intestine, bile comes, if you got fat, to emulsify it. And these other things come from the pancreas, enzymes we'll talk about, to try to further break it down. Because remember, you got to do everything right there, and you want to do it early in the duodenum. You'll continue it in the jejunum and ileum, but you want to do as much early, because like I say, once it gets into the large intestine, that's it. So you keep working on it, keep working on it. Then you get absorption, okay, of these nutrients. Then the absorption then is absorbed into the bloodstream if it's water soluble. Water soluble will be carbs, the amino acids, nucleic acids, things of that nature. But fats don't. Remember, fats go into the central lacteal, which ultimately goes into what? The intestinal trunk, which goes into the cisternae chyli, which then goes into the thoracic duct and into the bloodstream at the left subclavian vein. Okay, so then it gets into the bloodstream. But ultimately, all of your stuff, then you want to get into the cells to do metabolism, to get that out of it, if you're going to metabolize it. Now, the metabolism is only if you're going to burn it for energy. Listen to me. The metabolism is if you're only going to burn it for energy, which would be your proteins, carbohydrates, and fats. But fats, yes. Carbs, yes. Energy. Proteins. Let's hit this real quick. Proteins are your body. So you really absorb amino acids to make proteins in your body. That's what we call assimilation from ANP1. You're taking a cow meat and making it into human proteins. That's what's happening. So what you will do is, inside the cells, you will reassemble those. So when those amino acids go into a cell, inside the cell, they'll be reassembled to make proteins, because that's part of your body. Now, if you're in starvation, is the only time that you're going to use what? Amino acids for energy. But amino acids should be used to make proteins for growth and repair. What did I just say? For growth and repair. Your last resort energy is to use amino acids. It's kind of like if it was winter and you were in Alaska, way away from everybody, you ran out of firewood and you started consuming your house, tearing off the walls to get heat. Because when you start breaking down proteins, you're consuming yourself. So I just want to point that out, that though proteins can go through here and give energy, proteins, amino acids, you really don't want to use those. You're in a starvation mode when you have to start using those. Okay? When you use proteins, amino acids for energy, that's when you make urea. Remember urea in the kidney? That's when you make that. Urea is the breakdown product of when you start deaminating and making using that for energy. So I'm just giving you an overview of what you're looking at. The mitochondria, you need oxygen. That's where you get the lion's share of your ATP, 32 ATPs. Glycolysis, you only get a net of 2. Remember, net means to subtract. You're doing a lot of steps and you're getting a lot of energy. So you can do the 100-meter dash with your muscles doing this, 100-meter dash. But to do that 26-mile marathon, hey, you're going to have to use this. But remember, to use it, you have to have this hooked to the electron transport chain, which is within the mitochondria too. Certain drugs and certain poisons can uncouple that. Like I mentioned, cyanide. So that gives you kind of why you're doing this chemical digestion. That gives you. So listen to this video a couple of times if you don't know the overview. See, I like to get you to the forest, then go into the trees. So you see the overview. The overview. Okay? So now that the overview's out of the way, and if you've forgotten it, go back and study it again. Listen to then I'm going to start a new one to get into the mechanics of the digestion with the enzymes.